watched the preview of Osmosis Jones last night. Yeah, I have. Yeah. I'm not gonna lie. I'm sorry, you look fresh.
It's a banger.
I was like, could I possibly watch this entire thing? Maybe. If you guys get above a 92% average on the Block 5 exam, which is their psychology, I'll watch it.
How about we watch it?
Yeah, you guys watch it.
We watch it. What was wrong with it?
So in terms of like style of humor, it's like, I mean, how would you classify it? It's disgusting. Sophomore. I'm not mad at the animation, like that's fine. But and then I like according the preview itself, it didn't give me a good idea of like what the actual threat was, but I gather that the main antagonist, Thrax, is not like Bacillus and Tracis, but it's some virus. I need to watch more, I guess, just to find out that part. But yeah, it's like super misleading. Now, Thrax is a cool name, but it's like it makes you think that it's like anthrax. But I think it's virus. So yeah, we'll continue. We'll do more exploration and deep diving into Osmosis Jones later. But I won't go to full screen, but let's just watch this. Well, two cool little videos. This first one is showing phagocytosis. So what we're looking at here are just like a bunch of bacteria, right? And just looking at, and I have my sound muted, I don't know if there's narrative at all, but just look at kind of like, I don't know if this is real time or sped up, it doesn't say, but yeah, just going to town and eating those things. And then there's some kind of, I don't know, interesting animations that show this rolling surveillance immune system that's coming across these bacteria. It looks like it's kind of vacuuming them up, but what I think that's supposed to be depicting is that you have those chemical motifs and signatures coming out of those bacteria that we're going to be talking about today that are triggering some of these immune cells and giving them signals that, yeah, you need to be engulfed, you need to be processed, you need to be presented to other immune cells. And once it gets engulfed, it's going into this phagosome, this organelle, specialized organelle in the middle of those cells that degrades it, chops it up into little pieces, and ultimately presents those pieces on the surface. So that's kind of what's going on when you have one of these professional phagocytic cells destroying all this bacteria and then chopping them up and presenting them on the cell surface. So that's one video. This other one is super short, but it's kind of impressive. So we were talking about eosinophils a little bit and what their function is. Do you guys remember what one of their main functions is? Yeah, they can process parasites, specifically helminths, these multicellular worms. So they're still tiny, like this little worm that we're looking at right here, it's tiny. I don't know if, I mean, you can kind of extrapolate from the size of the cells nearby it, how big it is, but maybe it's larval. Regardless, it's not going to be phagocytized, right? It's way too big for a cell to come by and engulf it. So, hence, eosinophils coming in, doing their thing. There's obviously some recruitment going on, some activation of pile-on effect. Lots and lots of immune cells are piling on, and they are pumping that poor worm full of cytotoxic enzymes that are destroying that worm. So that's how, like one of the arms of the immune system, one of the tools it has to take down these big multicellular pathogens like helminths. Kind of cool, right? Neat. Yes, sir.
What's that term again where they kind of like cut the head off the bacteria and hold the head up for the antigen like presentation or?
Yeah. Yeah. So when a phagocyte encounters a bacteria or a virus or like a unicellular protozoan or yeast, something small, something that's able to engulf, when it gets engulfed, it's going to be degraded. It's taken up in endosomes. Those will, and we'll talk about this today too, but those endosomes will become increasingly toxic. They'll be acidified, there'll be proteases pumped into it, things that destroy and kill that pathogen. Let's say it's a bacteria. After it's destroyed, it literally gets disassembled by specialized enzymes called proteases. they break that down, they break that bacteria down into little pieces, and then those pieces get taken up into other trafficking vesicles. They get put on the surface along with another presentation molecule called major histocompatibility complex, or MHC. So that's how antigen presentation works, and that's exactly what we're going to be talking about today. Yep. Okay, so you guys want a few review questions from yesterday? Cool. All right. Let's see. Alright, so we left off talking about natural killer cells. I didn't put these in order, but these questions were just generated from information on the slides. Put them in a multiple choice format with 4 options instead of 5, but close enough. And then a very lazily animated correct answers on there, but at least you get a little bit of a sense of multiple choice formats. So let's take a look. All 5 of these questions are about neutrophils first. Know that in IPAP, we strongly discourage exam questions from being negative stems, so you're not really supposed to write this on exam, you're allowed a couple, so it's not impossible, but we really don't write these. But for the purposes of review and practice, we can do whatever we want, right? So, first question, what do you guys think? Which of the following is not a characteristic of neutrophils? Long-lived circulating for weeks, that's right. How long do they typically last?
3 days.
Yeah, up to 3 days. Sometimes 10 hours or so, and then they die on their own, even if they don't encounter pathogen. They're very short-lived. And are they typically residing in tissue or are they circulating? Circulating, yeah, in blood. What percentage of circulating leukocytes are neutrophils?
40 to 70.
40 to 70. So what's the most prevalent one of those cell types we talked about in terms of leukocytes? Neutrophils, yeah, they are the most prevalent. And then, sorry, I might ask you some more detailed questions on these. What are leukocytes? White blood cells, exactly. Okay. What are nets?
It's like a sneeze.
We think it's number 3? Yeah. Decondensed chromatin, DNA, histones, granular contents. Yep, exactly. Primary function of neutrophils, this font might not be big enough for you guys in the back. Can you read that? Small font? Okay, cool. Good eyesight. Primary function of neutrophils in the immune system is what would you say? Phagocytosis and killing of pathogens. Yep, especially bacteria. Good. And then the last one, what term is used to describe the process of neutrophils releasing their DNA? Yeah. Netosis, that's right. Yeah, very good. So those are the right answers that are bolded. Next set of cells, natural killer cells. What type of immune cells are they? Yeah, cytotoxic lymphocytes. Are they innate or are they adaptive? Innate. Yeah, the other lymphocytes are adaptive to B cells and T cells, but not natural killer cells. And what about these? If the denominator is lymphocytes, not leukocytes, but just those lymphocytes, what percentage do they typically represent? 5 to 15%, very good. Key feature of natural killer cells is their ability to target, ability to kill target cells without what? Right, without prior sensitization, which is how T cells kill their target cells. They need that prior sensitization and antigen presentation. How do they determine whether or not to kill a target cell? We'll talk about this a little bit more today too. They do. They do kill everything, but there is one of these answers that is
number 2.
Number 2, exactly. So a target cell, let's say a target cell doesn't need to be killed. It can persuade or convince, not to anthropomorphize it, but it can give a signal that says, don't kill me. And natural killer cells will respect and recognize those signals. So that's what an inhibitory signal is. When it's interacting with a cell and it's like, I think I'm going to kill you. That cell can say, no, no, I get it. So there is that mechanism. What is the name of the process by which natural killer cells kill target cells coated with antibodies? We're saying
Antibodies.
number 2, antibody-dependent cell-mediated cytotoxicity, exactly. Yeah, so antibodies are coming and coating that target, and natural killer cells are recognizing the FC portion of those antibodies and killing those cells. Very good. Granulocytes, lumping all these granulocytes together, we talked about 3 on that one slide. What were those 3 types of granulocytes on that one slide? Eosinophils, basophils, and mast cells, yes, exactly. Okay, so what type of leukocyte is primarily associated with defense against parasitic elements? Eosinophils, yep. Which is the characteristic of basophils? Histamine and heparin. Those granules are packed with histamine and heparin. Where do mast cells typically mature? Connective tissues. Very good. And which of the following cells expresses receptors for IgE antibodies? Mast cells. Major basic protein, MVP, is a cytotoxic protein found in the granules of which cell type? Eosinophils. Yes. Sure we got that. Oh, I'm sorry. For the mast cells, that should be bone marrow. Yeah, I think it was the basophils that mature in connective tissues. I'll go back and double check that.
On your slide, it is mast cells that are in connective tissues.
It is.
Yeah, and then bone marrow for baso and
Oh, okay. All right. My apologies. So just ignore that. Yeah, but everything else is right on there. Okay, macrophages, a really super important class of immune cells. They are best described as which one of these 4 characteristics, 4 descriptions? 2 large phagocytic cells found in tissues. Primary function is what? Phagocytosis, professional eaters. They're known for their ability to present antigen. This is such a dumb question. This makes them what type of cell? Antigen presenting cells, APCs. How long do macrophages typically live? Weeks to months. Yep, they are longer lived immune cells. And then besides eating and presenting antigens, they also play a role in which of these? Yep, tissue remodeling and wound healing. Good job. Okay, dendritic cells. I think this is the last one. Nope. Dendritic cells. Extensive review. They're primarily classified as what kind of cells? What's their primary function? Number 3. What else? Anyone think it's anything other than 3? What do they look like? What's the morphology? They have all the tendrils. What are those tendrils doing? Yeah, sticking them in little nooks and crannies and sampling in order to do what? Grab stuff and present it. Yeah, so their primary job, they're specialized, form follows function. They're designed to sample and pull in antigens, process them, and then do the little APCs. Key role in dendritic cells is to bridge the innate and adaptive response. Yes, that's what antigen presentation does primarily. Dendritic cells strategically located to perform their surveillance function. Where are they usually located? Barrier tissues, where they would be exposed to those antigens, exposed to those pathogens. What do dendritic cells do after they phagocytize a pathogen? Break it down, exactly. Yep, antigen presentation. So a lot of these questions are kind of redundant, but that's okay. Activation of T cells by dendritic cells is crucial because Yeah, the other ones are definitely incorrect. And if, as you're taking exams, do use that thought process. If you're not immediately sure what the correct answer is, go through the other ones and make sure you can eliminate those. So you know, like, all these are incorrect, so that means it must be this one that's a little bit generic, a little vague, but that's the correct one, which shapes the type of immune response that will occur. Okay, and then this is the last one, lymphocytes. What percentage of the leukocyte population is comprised of those lymphocytes? 30%? What is the primary role of lymphocytes in the immune system? Yeah, and number 2, with the exception of the one that we talked about already, which is the natural killer cells, that they are not antigen specific. Which type of lymphocyte is responsible for producing antibodies? Which one? B cells, exactly, yes. Where do T cells mature? In the thymus. And where are they produced? In the bone marrow. Yep, exactly. So they have to migrate out of the bone marrow and mature in the thymus. What is the primary function of cytotoxic T cells? Yeah. Yeah, the name pretty much tells you they're killing those infected cells. Okay. Alright, so we'll drive on. This is where we left off yesterday. So today we're going to finish up the innate immunity. Again, it'll be like another slow-paced day. Finish up the innate immunity today. Kick off with adaptive immunity tomorrow. And I don't know if we'll finish it all tomorrow, but it's probably not. So I hope you guys don't mind the slower pace, but it just feels okay. Good. It just feels better to take our time and listen to it. Okay, so what are we talking about today? We're talking about the complement system, which has a bunch of different functions in removing pathogens and promoting inflammation. We're talking about cytokines, which are key signaling molecules, really, really elaborate, important molecules, not cells, just immunomodulatory signaling molecules that a bunch of cells produce. And then antimicrobial peptides, I actually don't have a dedicated slide for this, but we already talked about it. You just need to know that these are peptides that are found particularly in mucus and secretions that will destroy bacteria in particular by inserting into membranes and causing them to lyse. So we talked about that previously. Okay, starting off with a little chart here, we see our lymphocytes, or I'm sorry, our leukocytes broadly, these different classes of leukocytes that we've talked about. Platelets are not leukocytes, but they might produce some inflammatory mediators. Tissue cells are certainly not white blood cells, are they? But that doesn't mean they don't contribute to this. They're citizens of the body and they have a responsibility. It's kind of like if you see something, say something, right? You don't have to be a professional immune cell in order to recognize that there's something going on and let the rest of the body know. So tissue cells too will secrete some of these cytokines and signaling molecules. But primarily, we'll talk about cytokines that are produced by these lymphocytes and then complement, which a bunch of cells also can produce. Start with complement system. So complement, these are a network of plasma proteins that are synthesized mostly in the liver. That's where a lot of them are manufactured. What type of cells do we find in the liver? Hepatocytes. Yeah, exactly. So those hepatocytes are actually producing a large quantity of these complement proteins that are secreted out, but other cells do as well. And their function is to complement, boost, or enhance, can be used synonymously, the function or the ability of antibodies and phagocytes to clear microbes. They do more than that, though. They also promote recruitment and activation of other immune systems, or immune cells as well. So multifunctional complement system. They are also important for both innate and adaptive immunity. And this is kind of an interesting micrograph where you can actually see complement doing its thing in terms of creating pores on liposomes. So these little dots right there That's pieces of complement that are forming complexes on those target surfaces. And there are probably 3 or 4 different types of complement that make up this pore structure, and then one, it's called C9, that actually forms a cylindrical stem that forms a pore, like a hole in that target membrane. So what that does is basically rupture that target cell. This is called the MAC complex. And in terms of its multifunctionality, this is one of the key functions it has, this membrane attack complex that was just shown on that previous slide. So this figure is a little bit busy, but it captures all of these 3 main functions, and let's kind of just talk through them, what they are. First one, the MAC complex, isn't very detailed on this, but when complement is produced, and when that complement binds to bacterial cells, for example, or some other target, it forms those pores, just like we saw in that other cell, and it leads to their lysis. That's all MAC is. We can get into much more detail, but I don't think it's worthwhile. Just know that that's what MAC complex is, something that directly lyses bacterial cells. Another function that it has is to recruit and activate other immune cells. So that's kind of what we're seeing here. This green molecule right here, that would be complement, those little pieces of protein that are sticking to bacterial cells. But let's say they're not able to form a MAC complex. Just sticking to them alone is useful, because what that'll do is it will draw the attention of some immune cell, like a macrophage or something like that. And those immune cells will have receptors on their surface for complement receptors on the cell surface. So that's what those orange cylinders are. Those are complement receptors that are recognizing that and allowing those immune cells to secrete cytokines, which we'll talk about in a second. Those cytokines are responsible for drawing out other immune cells. This will also activate mast cells. Remember those electron-dense, granule-heavy granules inside those mast cells? Those granules will be released, and those too will be responsible for recruiting additional immune cells. So those complement factors that are coming out and the cytokines that are being released will cause these immune cells to be recruited, coming in from blood vessels, so a lot of them will be neutrophils that are following that trail. And those neutrophils will extravasate through those endothelial cells in those blood vessels and come into that area. So in the tissue, of course, you have tiny little blood vessels and capillaries that serve that tissue. And recruiting these immune cells from those capillaries is really, really important. So they play a major role in recruiting and attracting those as well. The last function is kind of also diagrammed out here, where you're coating those bacterial surfaces or whatever other pathogen you might be encountering. You're coating them with complement, and that complement is recognized by phagocytes. And those phagocytes have a much easier time engulfing bacteria if they're coated with complement. That process is called opsonization. So complement is one of the most important opsonins in the body. Another really important one that we'll talk about later are antibodies themselves. That's one of the functions of antibodies. So antibodies and complement both serve to coat targets and make it easier to be phagocytized by things like macrophages, neutrophils, dendritic cells, and things like that. Okay, with cytokines, this is a very complicated system. And we'll see that in a couple slides, but the main function of cytokines is to modulate immune responses. This can be done by stimulating immune cells to react. It can be done by suppressing them and telling them that the threat is over and you need to calm down and deactivate and de-escalate. They also have a role in signaling that progenitor cells need to differentiate, like stem cells or undifferentiated granulocytes. So all of this is important in orchestrating and coordinating the immune response. They're secreted not just by immune cells, but they're also secreted by non-immune cells, importantly. Those endothelial cells, fibroblasts, epithelial cells in particular, any of those cells can secrete cytokines and contribute to this overall messaging effect. These two would have no effect were it not for their ability to bind to specific receptors on cell surfaces. So most cells are going to also have cytokine receptors on them as well. And in turn, let's say on this cartoon here, you see like any generic cytokine producing cell, could be anything, right? It receives some sort of stimulus. Let's say it's a threat. It senses that there's a bacterial cell nearby. So that is stimulus. It starts producing cytokines, like let's say pro-inflammatory cytokines. And those cytokines are wafting around in that immediate localized area, alerting other cells that there is a problem or some sort of dangerous hazard in that area. But those receiving cells will not receive the message unless they have that receptor, and they do. Receptors are common in most cells in the body. And then those target cells will do whatever they do in response. But it's a process that requires upregulation of genes in response to those signaling cascades. It happens on a very quick time scale so that they can respond very quickly. And for this activation, when cytokines are being produced, it's not always necessarily with the intended audience being neighboring cells. It could be a self-activation, so you get these positive feedback loops where your own cytokines are, you're turning yourself on. That's called autocrine signaling. So like IL-2, for example, is a type of cytokine that is released by a single cell, and it's received by receptors on that same cell that produced it in the first place, and that serves to amplify its whatever effect it's going to have. So that's an autocrine effect. A lot of times, if you're just notifying your neighbors, that's the paracrine effect, paracrine stimulation. And finally, if those cytokines drift out far distances, that's an endocrine effect. So all of these factors play a big role. There's a really, really interesting research group in Harvard that studies the complexity of cytokine signaling. Her name is An Kui, who's the lead of this lab. And if you ever want to watch like a really good, or listen to a good podcast, she's done a bunch of good podcast interviews, but she calls it the cytokine symphony. And that is an appropriate term because it alludes to the fact that you don't just have lots of different cytokines. There are like well over 250 different types of cytokines in the body, and they all have different functions and they do different things. But the amount that's released is important, and the timing of when they're released is important. And the way the message is received by cells nearby and far away, that's all important. When the suppressive signals kick in is super important. And if you don't have those suppressive signals, you get into these really dangerous situations where you have an uncontrolled stimulation of immune cells. You get into problems like serious systemic inflammatory response syndrome. It can lead to all sorts of problems, but that can be a fatal condition. Yes, sir?
So would this also be involved with autoimmune? Like they're attacking, like it's not recognizing that it's a bad, like a self?
No, so autoimmune is kind of, I think that's more like when you're failing to recognize signals appropriately. This is more like you're not able to control those signals. They're doing what they need to be doing, but they're doing too much of it, and you don't have a way to rein it in. So I think on the next slide, yeah, there are some important, so I said there are like 250, I just handpicked some of the most important ones for us to talk about. But like, take for example, IL-10. Its function is to down-regulate the cell-mediated response. So we're talking about CD4 cells and CD8 cells, cell-mediated response. Without cells or cytokines like IL-10, these T cells will just go to town and do a lot of damage. So you need to have that, you need to pump the brakes and have some of these anti-inflammatory cytokines do their thing. Let me go back and just hit those categories real fast before we go to that slide. So we do kind of break up cytokines into a couple different categories. Interleukins, interferons, and tumor necrosis factor. These are primarily going to be involved in stimulating and activating and also suppressing effector cells. Chemokines have a slightly different role. They do a lot of recruitment of immune cells. So that is accomplished by leaving a little trail of these molecules, trail of chemokines. So immune cells can pick up on a gradient and follow as that gradient increases, and it directs them and guides them straight into the area of injury that they need to go. So it's a little bit of a different function. Interleukins play a big role in communicating between different immune cells. macrophages communicating with dendritic cells, etc. Interferons, particularly interferon gamma, is super important in sensing and communicating viral threats. It's an antiviral role. And then tumor necrosis factor, particularly tumor necrosis factor alpha, TNF alpha, plays a big role in stimulating an immune response and causing cells to receive that death signal. So if a cell is infected with a virus, it's an intercellular pathogen, right? It's going to be needing to be targeted for apoptosis. And again, apoptosis is a desirable effect because it's a nice, clean, orderly way of destroying that entire cell. It's not ruptured all at once, it's kind of like systematically shut down and neatly packaged and then degraded by other cells. So apoptosis is a great way to go, nice, orderly elimination of that threat.
You said most cells have the receptors to accept the cytokines?
Yeah.
What's an example of a cell that wouldn't have those? It would be like alert others.
We don't know a single cell type that wouldn't have some sort of chemokine receptor. There may be, though. I'll look into that and see. Yeah. Yeah. Okay, so in terms of some of these processes, and some of these are very interlinked and closely associated, but let's just talk about a few of these effector functions that different cytokines cause or effectuate. IL-1 is a super important one. You probably won't see it much, but occasionally we might talk about interleukin-1. One of the subsets, a cleaved product of interleukin-1 is IL-1 beta. And that particular promotes fever conditions, the release of pyrogens, diapedesis, the extravasation of neutrophils and other white blood cells into infected, inflamed areas, inflammation itself, and it also activates macrophages. TNF alpha, super, super important when we just talked about it, contributes to apoptosis, also ensures that macrophages stay activated, which can be problematic. Can lead to septic shock at high levels. So we have a slide towards the end when we talk about dysfunctions of the immune system that will talk about how this sort of leads into an uncontrolled cascade, but TNF alpha is implicated a lot of the time in those really, really dangerous situations. IL-6 is super important. I don't know why it says by the liver. Most of these are synthesized by the liver. But activating lymphocytes, activating T cells. IL-2 is not a pro-inflammatory one, but it does promote proliferation in T cells. So when a T cell is responding correctly, antigen-specific to a pathogen, it will send signals that ensure those T cells are amplified and proliferated and expanded, those populations. IL-10 we already talked about has a suppressive effect, anti-inflammatory, super important. IL-12 promotes a specific type of T helper cell response called the Th1 response. Don't worry about that yet, we'll talk more about that tomorrow. But it also activates natural killer cells. Interference, already mentioned, antiviral effect, promotes fever, promotes cytotoxic T cells and B cell activation. And then interferon gamma, interferon gamma, a different type of interferon. Okay, so this slide is busy, I know it's horrifying, but don't worry, it does have an FYI up here, right, so you don't have to memorize it. It's just meant to illustrate and help put all this together if you're wondering how this works. So first, let's just look at the timeline here. We're looking at what happens immediately after you get some sort of injury or insult. So let's say something breaks the barrier of your skin and you get bacteria released locally into the skin. What happens immediately after that? You start getting recruitment of different immune cells. PMNs, you guys remember what those are? Those are neutrophils, yeah. Polymorphonuclear cells, PMNs, macrophages, dendritic cells, neutrophils start piling on into that area. Looking at this legend here, these arrows, if there's a big pink arrow like that, it means there's an effector function. So like a neutrophil with a big pink arrow heading towards the bacteria, that means it's gonna do its thing, which is what? What are neutrophils gonna do to this bacteria? Yeah, it's going to destroy destroy them, engulf them and destroy them. So they're coming in. If it's a purple arrow, it means it's activating. So that's kind of the whole point of this slide, is to look at all the ways that these cells are communicating with each other and activating other cells in order to have an effect. We talk about a dendritic cell. This dendritic cell is doing its thing, so it's taking in some bacteria, it's processing them in the middle of their cell, these lysosomes, storing them, processing them, presenting them on the surface. And then these arrows, these purple arrows, are showing all the different cells that it's presenting this antigen to, antigen presentation. So it's communicating with B cells, it's communicating with other dendritic cells, natural killer cells. And in the process, we're looking at the types of cytokines that are being released. We talked about some of these a little bit. Interleukin 12, TNF alpha, pro-inflammatory cytokines that are going off and activating other cells. We have recruitment of TNF alpha doing its turn, recruiting other immune cells, neutrophils coming into the area. This sign right there, theta, this is an activated macrophage. We see that a lot, but that's all it is. It's an activated macrophage that's being recruited into the area. And then it's doing what APCs do, right? It's recognizing those threats, it's presenting them on their surface, and it's telling T cells in particular that there's a threat, so you can start kicking off this adaptive immune response. This happens on that same timeline that I mentioned a couple days ago. Within 4 days or so, increasingly so, that gradient becomes more, it shifts away from the innate immune system into the adaptive immune system. So increasingly we see cytotoxic T cells, helper T cells, and some B cells starting to take a bigger role. So again, like the whole point, you don't need to memorize this, just kind of try and understand this a little bit. And what else? Just one other thing, because we were talking about inhibitory signals and how important they are. This green hashed arrow right there would be like an example of IL-10 having a suppressive effect, where you start dampening that response, start putting on the brakes a little bit. One other thing though, let's say this injury, that penetration takes place in your forearm or hand or whatever. This handoff, this presentation for antigen presenting cells, there will be some communication locally, but they need to migrate into a local lymph node in order to start activating those T cells. So here you can see these cells starting to move into lymph nodes so they can interact with T cells. That's where most of those T cells are going to be. Okay. So, yes, sir.
I noticed that all the purple arrows are pointing backwards to kind of activate something, except for 2. I was wondering how like the CD4 activates the neutrophil if the neutrophil comes first.
Yeah, that's a good question. So we haven't talked about the function of CD4 much, but those are the helper T cells. And what they do, they are also kind of professional, dedicated communicators and activators. So even though you might be thinking of them as like progressively down the line, downstream, they still produce a lot of cytokines and signaling molecules that will have upstream effects, suppressive and activating. So CD4 T cells will be interacting with CD8 cytotoxic T cells, they'll be interacting with B cells, they will be reaching back and giving messaging innate immune cells as well, oftentimes starting to shut them down and saying, back off of what you're doing. But yeah, it's not always moving forward. Sometimes you need to communicate back downstream or upstream as well. Okay, so super confusing slide, I get that. Think about it for a little while. We'll revisit it again, but hopefully it makes a little bit of sense about how cytokines control the immune response, like what a ginormous role they have in controlling this entire, or like orchestrating all of these things that need to happen. Okay, the rest of the lecture today we'll talk about some of these processes in a little bit more detail. We've already talked about them, but this will hopefully cinch those processes together and make them make more sense. But I've got 10:40, so this is a good break point. Let's take a 10-minute break and we'll start back up at 10:50. Well, I mean, it's kind of what I was
At least got that picture.
You have a very bad.
Is it like mostly like extravasation? And that's pretty much synonymous with diabetes. It's the same thing. Okay, well, I think covering that is something. That's why whenever I can, I try to bring down the quality of words because it helps you most figure out diabetes. Dia is like diabetes and basis is like jumping. So just think about that when you try to read it, just kind of squeeze into from a vocabulary and extravasation coming out as fast. Yeah. 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 Well, yeah, let me know if there's anything else that's you said.
S'mores. There used to be see that? I mean, it's Greek.
There's a trail right next to my head. Like a mile. What's the difference?
You said that when I was functions, some of these are multifunctional and also makes it think of it as
Your first two classes. I'm so jealous. The Georgia assignment sounds
Eating antibacterial, that's how they get around.
I think I've had like 3 conversations, if anything. I've been mostly operations units, so
only been just like
Okay guys, let's get going again. If you're looking at this slide and you're like, didn't we already go over this? You're absolutely right. I just wanted to make a couple more points on complement real quick that I forgot to make that I intended to make. And specifically about opsonization and MAC, you know, that pore-forming lysis capability that they have, the ability to lyse those cells. With respect to bacteria and this lysis function that they have, MAC. Gram-negatives and gram-positives, they have different susceptibilities to complement. And that relates to the fact that on gram-positives, they have a bunch of peptide ligands, right, on that outer layer. That's harder for complement complexes, MAC complexes, to penetrate through. For that reason, MAC is a lot more effective on gram-negatives. So I'll just throw that out there. It's kind of a nuanced point, but it is important. Particularly bacteria like Neisseria gonorrhea, Neisseria meningitis, it's very, very effective. Those gram-negatives, complement's very important, but lesser so with gram-positives. Another quick point that is kind of important to make with the lysis of gram-negative bacteria and other bacteria, when they lyse, a gram-negative bacterial cell lyses, that cell wall is released into the milieu, it's released into tissue or blood or wherever the infection is, right? We've talked about this a couple times, so I just want to kind of synthesize this point. What is in the cell wall of a gram-negative bacterium that is problematic when it's released? Yeah, lipid A, which has that endotoxin. So if you have a bunch of complement that is acting on a bunch of gram-negative cells in particular and causing them to lyse in an uncontrolled manner, just know that that can be problematic also. It can contribute to immunopathology, it can contribute to some problems for the host. It's a recurring theme, right? A lot of these immunological processes, they're necessary, but there's going to be some pain and there's going to be some collateral damage in the process. So that is important for that. And one other point on bacteria that are being targeted, like any bacteria that are being actioned upon by complement. We talked about capsule, and capsule being able to protect some gram-negatives and some gram-positives alike, right, forming that protective thick mucin layer. We said that capsule does what specifically to help bacteria?
hard to fake, covers the antigens and then sticks the stuff.
Yeah, a bunch of things, but it covers those antigens, prevents recognition. It also makes it harder for macrophages to engulf them, right? So, enteropsinins, like complement, like antibodies, it's still hard to attach to capsule, it's still sticky, but it makes it a lot easier if you're coated with one of those opsins. If you're coated with complement or if you're coated with antibodies, it makes it easier for these macrophages to come along and engulf those target cells. So I failed to mention that, but if you didn't have things like complement and antibodies, it would be much, much harder for these phagocytes to come along and do their job. It makes it easier for them to do that. So that's another important point I don't think I emphasized enough. Okay, so we were talking about processes. This is kind of an outline of what we're talking about. We'll start talking about recognition of pathogens, hinted that a little bit more, talk about the mechanisms of phagocytosis in more detail, talk about inflammation, well, maybe not the final pass the antigen presentation, but we'll talk about it in more detail. We'll talk about how natural killer cells do their thing. And cytokine production, I think we're already done talking about. We've said enough on that topic. So we'll start off with how any cells, immune and non-immune cells, recognize different threats. There are a couple different ways. One is pathogens themselves. They leave little signatures or patterns for these immune cells, these non-specific innate immune cells to recognize. So in the bottom here, This figure shows some of these generic patterns that can be recognized. Porins, you guys remember what might have porins on it? What are porins? Yeah, they are proteins in the cell wall of bacteria that form channels for nutrients to come in and out of. So those porins can be recognized just like broadly, just broad recognition. That's a porin, it's on a bacterial cell. I'm going to attack it. I'm going to destroy it. What about mannan? We haven't really talked about, but that's a carbohydrate that's in the cell wall of fungi. Double-stranded RNA. What might that indicate? Viruses, yeah. Some double-stranded RNA viruses, so that can be recognized. Flagellin, that's a protein that is in, what would you think? Yeah, flagellin is the protein that makes up flagella on bacteria, so that's super antigenic, meaning it can trigger an immune response and trigger recognition. Lipotycoic acids, zymosin, we haven't talked about zymosin, but that's another fungal cell wall carbohydrate, and these lipotycoic acids, that's in gram-positive cell walls. So all of these things are kind of broadly recognized in the manner analogous to what I was talking about, where, like, for me, for example, when I'm looking at you guys, just seeing pattern of you wearing a uniform, that's kind of it. It's nothing specific. So those are called PAMPs, pathogen-associated molecular patterns. You've got, what does GN stand for, do you think? Gram-negatives. And gram-negatives that have LPS, what's LPS? Lipopolysaccharides, LPS, which has, among its components, lipid A with the endotoxin. Yeah, so they'll recognize the LPS broadly. What's GP? Right, gram-positives will have another motif called LTA. What does LTA stand for? Lipoic acid. Yep, very good. Broadly, bacterial cells will have PG, which stands for peptidoglycan. Yes, which of those will have more peptidoglycan? Gram-positives have a ton, very thick layer of peptidoglycan, whereas with gram-negatives, it's sandwiched in between the inner and outer membranes, but still recognizable. Flagellin, we talked about. Double-stranded RNA, that's the DS, double-stranded RNA with some viruses, zymosin with fungi. And then we also have kind of like secondary indications that there might be problems or trouble. And that would be when the tissues and cells are being damaged themselves. You wouldn't normally find these outside of the cell. So when you see things like ATP, when you see uric acid, heat shock proteins, which are chaperones that are meant to make sure proteins are folded properly, all of those should be functioning inside the cell. So if you detect them outside the cell, that means there's damage. So those are damage-associated molecular patterns of DAMPs that are also recognized by immune cells or causes recruitment. The things that actually recognize the PAMPs and DAMPs are called PRRs, pattern recognition receptors, which are, yes sir? Oh, yes.
Yeah, I just had a question on still the good bacteria in our body, does that also have?
Yeah. Okay, so that's a great question. Particularly like in the gut, which we'll talk about tomorrow morning, which is like one of the hardest to surveil areas in the body. Your immune cells constantly have to be checking up on populations of bacteria in there. And it's kind of like, I don't know, I guess roughly analogous to immigration. You know, you are used to screening large quantities of people and saying you are good to go or you're not good to go, right? And in the gut, that's what those immune cells that are surveilling those populations do. They were trained to tolerate, that's the word we use, they were trained to tolerate friendly, beneficial commensals living in the gut and other mucosal tissues as well, have that same mechanism. So it's a great question. We won't go into a lot of detail, but if your immune cells do start targeting friendly bacteria, that can also lend itself to some bad conditions, inflammation, and overreactive, inappropriate immune response to things that it doesn't need to respond to. So that can be problematic as well. But they do have mechanisms for damping down those reactions to friendly commensal bacteria. Okay, so these pattern recognition receptors, these PRRs, they're on the surface of all these different cells that are interacting and being exposed to different microbial threats. They are broadly broken down into these classes called toll-like receptors. These are toll-like receptors or receptors that are on both on the outside of cells and on the inside of cells that are basically sensors for different motifs or patterns that indicate that there's a threat or a problem. You don't have to memorize these, but of these 10 classes of toll-like receptors that have been identified, roughly half will function to detect problems on the outside of the cell, and the other half will function to kind of be indications of, or indicators that there's a problem within the cell. So for example, like a virus, when that TLR, intracellular TLR is sensing that there is double-stranded RNA or something that's not normal in the cell, it will transduce that signal and the appropriate cytokines or other effects will be effectuated after that. Okay, so it's a sensor, it causes an appropriate response to that threat. That's the whole idea. And that kicks off all of that communication that we saw in that very busy slide a couple slides ago.
Are TLRs are always PRRs?
Yeah.
Are PRRs always going to be TLRs?
No, they aren't. TLRs are kind of the main sensors of that superfamily of receptors, of PRRs, and they're the only ones that we really need to focus on, but there are other types of PRRs as well. Yeah, it's an extremely complex topic, so I'm sort of narrowing down our scope a little bit, but it is important to know what toll-like receptors are. And this is another FYI slide, so don't freak out. It's just something that helps to kind of synthesize everything and put it together, but it's broken up kind of the same way as that last slide, where you have a top row here that has some of the TLRs that are associated with cell surface sensing, and then these intercellular TLRs. And if you've taken a rigorous class in cell biology before, you might recognize some of these other sensing cells, but like, for example, GRID1, NDA5. Those are all additional sensing molecules that their sole job is to pick up things that are aberrant or abnormal and transduce those signals right to the cell. But yeah, viruses, RNA, DNA, or GP, that's the glycoprotein, that's the receptor on the viral surfaces. That will trigger a TLR response. Gram-positives, their DNA. You might be thinking, isn't the DNA of the bacteria basically the same as the DNA of some eukaryotic cell? Not really. There are subtle differences. The DNA itself is the same, but it has a different GC content. It can be methylated differently. So there are slight things that your immune system can use to pick up on and notice that is bacterial DNA on the inside. And these are for intracellular bacterial pathogens, things like Salmonella, Listeria, Mycobacterium, but most are going to be extracellular. Peptidoglycan, liponucleic acid we've talked about, with gram-negatives, peptidoglycan probably to a lesser extent, flagella perhaps, which can also be on gram-positives. Fungal pathogens, zymosin, mannins, glucans, chitin, that can be immunogenic. And even with protozoans, we have a couple TLRs that we can recognize indicators or patterns that are associated with protozoans. Okay, in terms of processes, those are pattern recognition. This slide is kind of just telling you that different types of pathogens will elicit different types of immune response. It's sort of like an overview slide, but when you're talking about basic microbiology and we're starting to cover day by day different pathogens like influenza or strep or tuberculosis or malaria, just know that your immune system is recognizing all of these different pathogens differently according to what kind of toll-like receptor is sensing it in the first place and what kind of cytokines are being produced. So it's all a pretty elaborate thing, and that's why the immune response to different pathogens varies so greatly. Some different pathogens, the pathogen itself might not be causing a lot of the damage that's responsible for disease, but it might be tripping your immune system in such a way that a lot of the pathology is actually immunopathology. It's caused by the immune system itself. And it's also very important as we're talking in blocks 3, 4, and 5, if it's an intracellular pathogen, it elicits a very different response than an extracellular pathogen. So something like strep, that's a strict extracellular pathogen. It might be adhering to the surface of the cells, but it's never going to be invading inside of them. So it's not tripping off the same sensors, and it's not shaping the same immune response as some of these other pathogens. Okay, in terms of recruitment, we've talked about this a little bit, but you have the initial recognition of danger from PAMPs or from DAMPs. And from there, you have release of things like cytokines and other indicators that you need to recruit more help to kill this infection. So you get release of these cytokines and then a recruitment of cells from circulation, things like neutrophils. And this process is called diaphoresis, when these neutrophils that are flowing past through capillaries and the blood vessels are slowed down and recruited into that area. So depending on what class of blood is flowing in that particular vessel, what needs to happen is they need to start making some light attachments to those endothelial cells. That slows down their momentum. They continue to slow until they come to basically a stop at the junction of two endothelial cells. And then because of the way their cell is shaped, it's a very flexible cell, they can actually extravasate through those two different tight cells in that endothelial junction and make their way into the tissue. So that is diaphoresis. In the process of squeezing through those cells, what that does is it loosens up the tight junctions in between those endothelial cells. And that causes those vessels to start leaking a little bit, they become leaky. And that causes edema, inflammation, and some problems additionally, but that's what you have to do in order to get the help into that area. So there are negative consequences as well, but you gotta get those neutrophils into that area somehow.
And is that temporary leakage? Do they kind of go back? Because that edema doesn't last forever, so I assume that they kind of resort back to the tight junction.
Exactly. So these endothelial cells, they have different adhesive molecules that they produce, extracellular matrix molecules that are sticky, and if they get damaged or displaced, they'll produce more, so it'll close up and it'll heal itself on its own. But if this becomes out of control, it can be difficult to maintain blood pressure, basically. If you have like a rampant systemic problem with your blood vessels maintaining their integrity, that can become really dangerous as well. But otherwise, it should heal, and it'll heal quickly on its own. With phagocytosis, we talked about a couple of immune cells that are responsible and specialized for the process of phagocytosis. What are some of those immune cells that we talked about? Macrophages, neutrophils, dendritic cells, exactly. And the progenitor of macrophages and dendritic cells, monocytes, can also have some phagocytic capabilities as well. It's not as good at phagocytizing targets as fully fledged differentiated macrophages, but it can also be done. Okay, so what happens here, you have a bacterial cell. This bacteria has been, in this cartoon, it has already been opsonized by one of those opsonins that we've talked about. In this case, it's an antibody. So your body has had time to recognize and produce antibodies against this bacteria. The Fc region of those antibodies, which we'll talk about more, is being recognized by a receptor specifically for it to help that macrophage take it into the cell. That's all it's doing is an opsonin. It's helping the macrophage help it. That bacteria gets taken up into a vesicle called an endosome, and those endosomes are triggered. They recognize that there is something bad in there that needs to be destroyed. So when they're triggered, they start releasing all this really, really bad stuff. You got chloride ions, basically bleach that goes in there, hydrogen peroxide, free radicals, reactant oxygen species, a lot of bad stuff. Starts pouring into those. Some granules will start fusing with that compartment, which will become a lysosome. The proton pumps, these transmembrane proteins that are in these endosomes, they will start acidifying those compartments as well, so they'll start pumping protons across that gradient. And the pH will drop significantly. So all of those factors together will kill, at least that's what the intent is, they'll kill whatever bacteria or other pathogen is in those compartments. After that killing is done, now you have to rip everything apart. So enter the proteases. What are proteases?
Enzymes that break down proteins.
Enzymes that break down proteins, exactly. So if you hear suffix ACE, A-S-E, that's usually an indicator that you're dealing with an enzyme of some sort. You have nucleases, collagenases, elastases, proteases, hyaluronidases, enzymes that break down everything, right? So these degradative enzymes will come in and they will just shred those bacteria to pieces, and then they will put those pieces together in little vesicles to get exocytosed out of the cell. Exocytosis is sometimes referred to as cellular defecation. It's a little crude, but you gotta get rid of most of that waste, right? But what leaves is non-toxic. It's already been ripped to shreds, so it's not gonna cause any problems for the body. It's not going to elicit strong response, just getting rid of that waste. So it's a normal cellular process, not a big deal. Some of that shredded material will be used for antigen presentation. So those peptides, which are usually between like 18 to 30 amino acids long, will be loaded up onto specific molecules called major histocompatibility complexes. And then together, those will get trafficked to the plasma membrane of that cell and presented for all the world to see. This is my peptide, do with it what you will. Okay. And we'll talk about that in a second too with antigen presentation. With the inflammatory response, I should have put this after the diabetes slide, it would have said better, but with inflammation, good and bad, that's the point. For good, what this does is allow those really important immune cells into the affected area so they can clear those bacteria or whatever other pathogens. So that's important, and it's worth all of these bad things that happen as a result of that action, of the recruitment of those neutrophils into that area. You're probably already familiar with this, you've probably already all experienced inflammation at some point. This is a localized response usually, where you get these major signs. You get redness, which is called erythema, or something that's erythematous. You get heat locally. I'm not talking about fever, which is systemic. I'm talking about like when you touch that area, it feels hot to the touch. And that's because those blood vessels have dilated, they've widened, and that's what's responsible for a lot of these signs and symptoms, right? The increased blood flow is going to cause that extra heat. It's going to make that area red. It's going to cause some swelling because of the vascular permeability. So those connective junctions are loosened up by neutrophils coming into the area. That's called edema. And it's going to hurt. So unfortunately, you have the release of some of these molecules like bradykinins, prostaglandins, and others that will be binding to pain receptors and sending pain signals to your brain. There's probably a purpose to that too, but to let you know that there's a problem, I guess. But all this suffering is so that you can ultimately clear those bacteria. With fever, slightly different. We've talked about fever a little bit before, but fever is a systemic response to infection. It's elicited by molecules called pyrogens that basically are responsible for resetting your internal thermostat, setting it, kicking it up a notch or two. So hypothalamus controls that, this is part of your brain. And if you were to reset your internal temperature from 98 degrees to what we'd call a low-grade fever, maybe 99, 100 degrees, that's going to do a lot to suppress different pathogens. Okay? And if you kick it up to what we'd call a high-grade fever, that will affect it even more. So for every day you spend in misery with a fever, it's doing a lot of good in terms of slowing down that threat. Talk about this a little bit, but when you have elevated temperatures, what does that do to you, like on a cellular level? Eventually, what will it do to proteins?
Denatures them.
It'll denature them, right? So if you have denatured proteins, yes, that will hurt the bacteria, the viruses, and will kill them. But even for you, when you start getting into that really dangerous area where you have really high fever, your own proteins start functioning well. that causes cellular processes to start collapsing. So you lose cell functionality. If you lose cell functionality and you have significant cytopathology, entire tissues will give out. And in all of your vital organs, this is going to be problematic. So when this happens in your brain, when you start losing neurons in your brain, you start having serious neurological symptoms. So you start having seizures, you can fall into a coma, you can die that way. Your cells in your heart, cardiac cells, those can start malfunctioning and you can basically have a heart attack, cardiac failure, respiratory failure, all of these things, not to mention what can happen to your vasculature. So those can all give out. Yes, sir?
Now, since we're saying a fever is good for the body to get rid of the infections you have.
Yeah.
Should we therefore not talk about the fever and get it?
Yeah, so that's a great question. In modern medicine, we've only recently had the ability to bring down fevers with fever-reducing medications, right? People have different opinions on this, like when to actually bring down fever versus when to just let it run its course. You know, particularly with kids. I don't know how many of you guys are parents. I think a lot of you probably. But like, if your kid has a fever, most parents are going to be freaking out. They're going to want to put their kid on fever reducers really quickly. And that's fine, but just know that low-grade fevers, that's survivable, and that's the body's way of very quickly and adeptly and efficiently handling those pathogens. We've evolved to do it that way. It's an effective thing. We've been doing it since before we were humans. So if you can let it run its course, just know that there's a lot of benefit to those fevers as well. But when if they become a high fever and they're dangerous, you probably need to do something about it. Does that answer your question? Okay.
What's the line, like, when the bad for bacteria, and still okay for you, but start doing bad for you?
There's really no precise way to answer that. I don't even, a lot of clinicians don't like the terms low-grade and high-grade fever at all because it's a little hazy, it's fuzzy. But I know by gradations, every degree up is significant. Like, it's a big deal when you go from 100 to 101 to 102 to 103. Each one of those is important. But it probably depends on your own immune status, your own age, you're young and healthy versus frail, full of comorbidities, and the nature of the infection itself. That too is important. Yeah, very complex questions. Okay, inflammatory response, nothing new here, just a bunch of cytokines that are causing recruitment of immune cells into the area, diapedesis, vasodilation, sterilization, all of that stuff that we talked about, contributing to inflammatory conditions. Okay, super important. And then diapedesis again, where you have a neutrophil coming in from circulation in a blood vessel, needs to go down to a tissue, that's where all those chemoattractants are, so it's going through chemotaxis or traveling by following the chemical gradient, starting to slow down, roll, come to a complete stop, adhere, and then finally like squeeze its way in through that blood vessel. Okay. One more slide on antigen presentation. Just kind of, I think mostly we've talked about immunity a little bit, but now we want to formally understand what this function is, what this role of histocompatibility complex is. But let's go back for a second. We were talking about immune cells and we were talking about professional APCs. Do you guys remember what those professional APCs were? Dendritic cells are the very best, right? Ones with those tentacles that reach out. What other cells can macrophages, the multifunctional ones that do a little bit of everything. There's one more that we might not have even, in fact, I don't think we talked about, but B cells, yeah. B cells can also present antigen. We'll talk about that mechanism in just a second, but before I forget, this handy little acronym is one that you should I recommend that you remember that. DUM, dendritic cells, macrophages, B cells, those are your primary antigen presenting cells with no U. So these cells are specialized at taking up antigen through different mechanisms and processing it and putting it on their surface in order to basically activate the adaptive immune system by presenting it to T cells. So in this scan electron micrograph down here in the bottom right of your screen, this pink thing is a macrophage. And it's a big cell, like I said, the macrophages are large cells. It is interacting with a T cell down here, a nice, pretty, tight, small T cell. There's a lot of different receptor interactions going on here, not just the MHC reaction, but basically what it's doing when it's presenting antigen to that T cell is telling it that this particular antigen, I found this, and you need to respond and develop a specific response to it. So what's happening specifically, you have that peptide that is loaded up, and it is presented on this major histocompatibility complex, this MHC molecule. That happens inside the cell. It is placed in complex together, and then it's trafficked up to the surface here, where the T cell expresses a T cell receptor, a TCR. And that TCR will recognize that peptide that is presented to it, that peptide of 18 to 30 amino acids from that particular pathogen process. In terms of antigen capture, macrophages, of course, when they're engulfing and digesting bacteria, that's a slightly different process. They're completely ripping apart. With B cells, they oftentimes will take that antigen up through receptor-mediated endocytosis. They'll take it up, process it, break it down, and present peptides that way. But regardless, it all gets presented the same way on MHC. Okay, so those are our professional antigen-presenting cells. And those will express a specific class of major histocompatibility complex proteins called MHC2. So those are only found in APC. You don't find MHC class 2 on any other cells other than your dendritic cells, your macrophages, and your B cells. That is it. There is another class of major histocompatibility proteins called MHC class 1, and those are going to be on every cell in the body that is nucleated. So the only cells that aren't nucleated are basically like red blood cells, right? Your erythrocytes. So you don't see any MHC class 1 on those, but like everything else you do. And that is because they are specialized at presenting intercellular antigens. Pretty much every cell in your body is infectable by a virus. Probably a virus that can infect any tissue type, from your neurons to your hepatocytes to specialized pneumocytes, all of it, right? So that's why all nucleated cells, since they can become victims to intercellular pathogens like viruses, that's why they all need to present MHC class 1. In terms of where they hand this peptide off to, depending on whether you are expressing MHC class 1 or MHC class 2, you will have different target T cells to present that off to. If you are just like a normal skin cell or like an endothelial cell, for example, and you're infected with a virus, you're going to hand that off to a CD8 or cytotoxic T cell. If you are one of those professional APCs, dendritic cells, P cells, or macrophages, you're going to hand that off to CD4 helper cells. Okay, so that's important to note here. And then this cartoon here on the right is kind of showing that. This is showing like annual nucleated cell that unfortunately is infected with a virus. It's going to be processing that virus or capturing little pieces of it on its MHC1 complex and then presenting it at the cell surface so that it can be recognized by the TCR, the T cell receptor of the CD8 cell. Does that make sense? Okay. And then with an APC, again, these are your specialized immune cells that are either taking in antigens by receptor-mediated endocytosis, or they're phagocytizing them, they're engulfing them, and then they're processing that on a different molecule, MHC class 2, and handing that off to CD4 cells. Quick point there, in terms of MHC, for every person we have slightly different MHC that's expressed. So if you were to do like a tissue transplant between one person and another, that's one of the reasons why you get a tissue rejection. It's because your body recognizes someone else's THC as foreign, and you would mount an immune response to that. So it's you're probably aware of all the problems that are associated with like tissue transplants and organ transplants, right? Tissue rejection. One of the main reasons is because of MHC. It's different for every person, so you have to find a suitable person who has similar MHC. Up at the top, I don't get this on your slides, but I just put that there to kind of help you, but it's saying the same exact thing as in these bullets, that MHC class 1 interacts with the TCR, the cytotoxic T cells, and MHC2 interacts with CD4 T cells. Okay, almost done. With macrophages, we've talked about this a little bit, but this figure is really nice because it does underscore all the different functions that they have. And it also kind of reinforces how important receptors are. So it's like a Swiss Army knife of immune cells, but starting up here with cell activation, we've talked about some of these cytokines before, but those macrophages would have receptors for the cytokines. So if they're encountering pro-inflammatory cytokines like TNF-alpha or interferon gamma, they'd have a signaling cascade that would cause them to be activated when those are stimulated. For microbe recognition, we have toll-like receptors that we've talked about. There's a specific receptor just for LPS on these macrophages. Mannose receptors, these receptors, I don't even know what these receptors are, but they're designed to recognize patterns on microbes. Going at the bottom here, complement and Fc receptors for antibodies. We talked about those two components before. There's a word that describes both complement and antibodies. You guys remember what that word is? Like something that coats a bacteria that it opsins. Exactly. So macrophages will have receptors for those two different classes of opsins, complement and antibody, and that will facilitate uptake or make it even easier for them to engulf it. And in terms of antigen presentation, a whole bunch of stuff there too. Because they're nucleated, they have both MHC class 1, and because they're an APC, they have MHC class 2 on their surface. But only those APCs will have both. Other ones just have MHC class 1. And then you also have adhesion molecules like LFA and ICAMs. Those are important for forming tight connections with other immune cells when they need to convey those messages. So a lot of stuff going on, but this is why they're such incredibly versatile, adaptive immune cells. Very important. And last slide of today is talking a little bit more about natural killer cell cytotoxicity and how that works. So with natural killer cells, you are recognizing a different target, and in this case, you're recognizing the absence of MHC. When you have a virally infected cell, sometimes the virus will cause those cells to downregulate expression of MHC class 1 on the surface, with the intent being to kind of prevent antigen presentation from going on. When those are downregulated, T cells won't recognize them, so they won't have a way of seeing them. And that could be problematic, but that's why we have natural killer cells. It'll actually recognize the absence of that MHC molecule and kill those cells. So that's I'm trying to think of a good way to phrase that, but it's kind of like, whatever, it's going to kill it because it doesn't have those MHC molecules on the surface. I'll think of a better way of describing that later. In terms of how it kills, there are two closely related methods. In one of those methods, it forms an immunological synapse, a very close association with the target cell membrane. It basically fuses with the target cell membrane, and then it pumps the granules into that target cell. One of those is perforin, which creates a pore in that membrane, and then the granzymes are actually those molecules that will come in and start activating pro-apoptosis pathways in the target cell. Very similarly, you have what's called death receptor engagement. FASL ligand is expressed on natural killer cells. It will bind to its cognate receptor on target cells, and that too will cause a signaling cascade that leads to apoptosis of the target cell. Either way, both mechanisms result in apoptosis, so that's the killing mechanism for natural killer cells. Okay. And that's the last slide for the Innate community part of this deck. Any questions on this stuff? Maybe take some time tonight and look over these. I know you do have that exam going on, but we'll do some review tomorrow and make sure it all makes sense. But yeah, and we'll pick out the names that definitely makes sense. Okay, thank you guys. Let me know if you have any questions. I know you're done for the day, right, Jake? You guys are done? Okay. Well, have a great rest of your day and we'll see you tomorrow at 10.